Hello everyone, myself Sonia from 2nd MSc Biology, St. Mary's College, Thrissur. Here we are going to discuss about cell organelles such as lysosome, endosomes and microbodies. So let us first move to lysosome. Lysosomes are a vesicular, membrane-bound and enzyme-filled cell organelle, and it is mainly concerned with intracellular and extracellular digestion. First observed in liver cell as dense particle and termed as pericanalicular bodies, and it is isolated and named lysosome by Christian Dew in 1995. Present mostly in eukaryotic cells, and prokaryotes do not have true lysosomes. It's a structure of cell where we can see lysosomes. Structure and chemistry of lysosomes. Lysosomes are usually rounded or globular in shape and is surrounded by multivesicular bodies, smooth and coated vesicles and dense bodies. Lysosomes is a fluid-filled bag or rather an acidic compartment of the cell. It consists of an inner acidic matrix which contain highly powerful hydrolytic enzymes enveloped by a single highly dynamic limiting membrane, and it is mostly micellar with a lipoprotein composition. The membranes are glycosylated to protect themselves from degradation by lysosomal proteases. Lysosomal membrane contains transmembrane proteins. Proton pump help to maintain acidic medium within the lysosome. Why lysosomes called suicide bags? Lysosome membrane contains certain substances called stabilizers which make it stable and impermeable to lysosomal enzymes. For example, cholesterol, cortisol, heparin, etc. Certain other substances can destabilize the membrane and make it permeable to lysosomal enzymes, and they are called labilizers. Example: progesterone, testosterone, vitamin A, vitamin K, etc. If the cell gets damaged, then one of the lysosome bursts and release some digestive enzymes. The released enzymes then digest their own cell and ultimately the cell dies. Hence, lysosomes are called suicide bags of the cell. Functional variants or polymorphism in lysosome. Lysosomes are extremely dynamic and highly polymorphic structures, and they exist in different forms at different stages of their life cycle. The four functional kind of lysosomes are: first one is primary lysosome, secondary lysosome, telolysosome, and the fourth one is post lysosomes. Then, what is primary lysosomes? Primary lysosomes are the nascent or newly formed lysosomes, often called storage granules, and the secondary lysosome, which is mainly involved in intracellular digestive function. They are of two kinds: heterolysosomes and autolysosomes. Third one is telolysosomes and postlysosome. They are the aged or late stages of lysosome and formed when lysosomal digestion is partial or incomplete. The functions are intracellular digestion, dissolution of cell, role in gametes and fertilization, release of thyroid hormone, trigger mitosis, and bone resorption. Next is endosomes. Endosomes are primarily sorting intracellular organelle, membrane-bound vesicle formed by endocytosis, and it involved in the invagination of plasma membrane to the surround macromolecules. They are regulate they regulate the trafficking of protein and lipid among other subcellular compartments of the secretory and endocytic pathway. Endosomes are the integral part of endocytosis. It's a structure of the endocytosis in animal cells. There are three major compartments of endosomes. First, early endosome, second late endosome, and third one is recycling compartment of endosome. Early endosomes are seen close to plasma membrane and formed following endocytosis. An initial sorting station where budding of vesicle to different destination occurs. Light endosome located near to nucleus and send material for the enzymatic degradation. And the last is recycling endosome. Next is microbodies. Microbodies are small membrane bound enzyme filled cell organelles found in close association with endoplasmic reticulum, lipid bodies, etc. Discovered and named by Rodin in 1954. And the structure, usually rounded or elongated structures and delimited by a single membrane, filled with a densely granular, and their matrix is rich with enzymes. Microbodies are oxidative organelles and actively involved in the breakdown of H2O2 and fats, and also oxidation of amino acids, fatty acids, and uric acids. Classes of microbodies. Based on their morphological features and enzymatic compositions and metabolic roles, there are three classes of microbodies. First, peroxisome, microperoxisome, and third one is glyoxysomes. So, what is peroxisomes? Peroxisomes are self-duplicating spherical or dumbbell-shaped fluid-filled and membrane-bound bodies found in large numbers in protists, yeast, some type of plant cells, and kidney cells of animals. Consist of an enzyme-rich granular matrix delimited by a single unit membrane, and the peroxisomal membrane is relatively thinner and freely permeable to amino acids, uric acid, alpha hydroxy acid, etc. Peroxisomal matrix is rich in enzymes such as oxidases and catalyses. It's a structure of peroxisome with a lipid bilayer, crystalline core, and transport protein. Two major classes of peroxisomes. First one is animal peroxisome and the other one is plant peroxisome. Animal peroxisome are mainly concerned with the oxidation of amino acids and uric acids, especially numerous in liver cells and kidney cells of vertebrates. Also found in brain, small intestine, cortex, testes of vertebrates, etc. Functions: generation and breakdown of H2O2, beta oxidation of fatty acid. Next is plant peroxisome. They are most complex type of microbodies found in the photosynthetic tissue of green leaves with chloroplast and contain the enzymes of glycolate pathway of photorespiration, uric acid oxidases, catalysis, etc. And they are actively involved in the metabolism of amino acid, purine, H2O2 and photorespiration. Next is microperoxisomes, and they are the special type of peroxisome found in mammalian tissue other than kidney and liver, and they are characterized by small size and absence of crystalline core, presence of catalytic and oxidative enzymes, and they have numerous slender connection to smooth endoplasmic reticulum. The last one is glyoxysomes. They are highly specialized plant microbodies. They are concerned with the conversion of fatty acid to carbohydrate in the glyoxylic acid cycle. They are roughly spherical organelle consisting of a protein matrix, crystalline core delimited by a single unit membrane. Thank you.